For this assignment, we are going to be looking at the learning objective, learn how to do a dihybrid cross on a Punnett square. But before we get to that tricky stuff, we're going to go over some of the basics. So to start, we're going to learn about a basic Punnett square. So what a Punnett square is it's a fancy way to predict the traits that you're going to get from your parents. So this is what a normal Punnett square looks like. We're going to go over some of the vocabulary that you may encounter during this. So a genotype, which is one of the words I highlighted, is a gene of an organism. It's a specific trait that you use two letters to represent. A capital letter represents the dominant form of the gene allele. A lowercase letter represents the recessive form. A phenotype, on the other hand, is the physical appearance of a trait on the organism. So the difference between a genotype, when you do a Punnett square, is the genotype is the letters that are represented, while the phenotype is the trait that's represented. So an example of a trait would be long hair. Another word that's important is something called homozygous. So homozygous means pure. So when you have a homozygous, you can look to my right and you see big R, big R. This is different from heterozygous, which is a hybrid, meaning you'll have one of each, a big R and a little r. So when you do the Punnett square, let me scroll down, you start with your blank window. Say you have two parents, big T, and little t, little t. This is referred to as a monohybrid test cross. So when you're doing this, you're going to put one of the parental genotypes, big T, on the side right here. You then put the other parental types at the top, right here. Then you go down, you do 1, 1 gives you this. You take the second one, and you put it in this square. So when you get to the end, you're going to have something called a genotypic ratio and a phenotypic ratio. So recalling back to what we've been already discussing, genotypes has to do with the letters, while phenotypes has to do with the trait. So for this, you see that the genotype big T, little t represents 50% or 2 out of the 4. Little t represents the other 50%. This is different from the phenotypic relationship because the phenotypic relationship ratio uses tall and short. So 50% would be big T, little t, big T, little t. That's tall. 50% would also be short. The allele that codes for short is the recessive little t, as noted right here. So now we're going to look at a dihybrid cross. So we're going to go back to this page right here. So a dihybrid cross is a little bit trickier than a monohybrid cross. With a dihybrid cross, you have two sets of different traits. So you can have a big S, little s, and a big Y, little y. This can be a little tricky to figure out what exactly goes in the Punnett square. You also have to realize that the Punnett square is going to be a little bit different than it was originally. There's going to be more boxes. So in order to find out what your parentals are that go up here and what goes down here, you have to do a mini sort of Punnett square off to the side. So you take big S and big Y. You know that those compare together, they go right here. You then take little s, big S, and little y, they go right here. You repeat that for the other two. You take the little s and the big Y and you put it right here, and then the little s and the little y right here. Since this is being crossed with itself, you're going to have the same parents right down here. So we think back, we go big S, big Y, big S, little y, little s, big Y, little s, little y. So each of these traits are going to code for something different. So S can be for size or shape, and Y can be for color. So once you do the cross out, you still follow the same way that you did back during the other one. So we have to think back. So you take the big S, big Y, they go right here. Then big S, little y, right here, and you follow the same sort of pattern. So this will result in spherical, which is big S, and yellow, which is the Y. But for these ones, since there's a little or recessive S or a little recessive Y, that's going to code for something else. So big S would be round. Little y would mean green. So you see right here, you have one of those options right here. So the result of this cross would result in a phenotypic ratio. So think back to what phenotypic means. It means the color in the trait, or the expression of the gene. You would get a 9 to 3 to 3 to 1. So breaking that down, because that can be a little intimidating, you see that you have 9 big S, big Y. So big S, big Y, 1, 2, 3, 4, 5, 6, 7, 8, and 9. The 3 represents you have a big S and a little y. So that represents the green. So one green right here, one green right here, one green right here. So this is still round because it has the big S. The other 3 represents the recessive S. So that codes for the wrinkled in this case. So you have wrinkled, wrinkled, wrinkled. But since there's a uppercase Y, which means it's dominant for the color yellow, it will produce a yellow wrinkled. Lastly, you have the 1. This represents the homozygous recessive. So it has 2 recessive traits. Think back to what recessive means. It's the lowercase. So here you have little s, little s, little y, little y. So it's recessive for both the traits. So that's why it's green and wrinkled. So these dihybrid crosses can be a little trickier just because you have to figure out what the parentals are up here and what all the ratio means. But if you think back to our normal monohybrid, it's the same steps. You put the parents here, and then you cross them. And then that's it. We can figure out what genes you're going to get from your parents and the ratios that they will record in.